Yeah.
Okay.
Oh, did you want me to start again? Oh, man. Okay, let's see. Replay, please.
No, it's fine.
Okay. Good morning, everyone. My name's Orenda, and I'm here to give a little presentation on how I integrate ABE or the ABE curriculum into my classes. And I just want to say thank you to Amgen because they've been supporting me since Sam was born, right? Since I started the program long time back, and I'll talk about that. And I just want to say thank you to Karen and Sam for inviting me, and Wendy isn't here, but Wendy has always been supported our program. So thank you, and thank you to Robert because he's videotaping this. So thank you. All right, but real quick though, can I just ask who's a new teacher? Anyone? All right. Okay. Of the new teachers, are you guys teaching biology or Living Earth or AP Bio? Both. Both. Okay. So, but most of you guys are seasoned teachers. Okay, good. Is this your first time to do ABE, or no? Okay, so you guys have you've been doing this. So you guys should be here instead. You should be presenting, right? So instead of me. So anyway, all right. So So I'm, probably you can tell, I'm from Crescenta Valley High School. So Crescenta Valley High School is part of, it's in La Crescenta. It's part of the Glendale Unified School. Anyone from Glendale Unified School? Where?
I went to Crescenta Valley.
Oh, you went to CV? Woohoo! Yes. Go Falcons. Yes. All right. So I think I'm going to head over here. So we are part of the Glendale Unified School District. Okay. So these are classes that I teach. So Living Earth, or what used to be called biology, AP Bio, biotechnology, and science research class. So our bell schedule is, well, since COVID, we had block schedule, but starting next year, we're going back to 6 periods per day, which is brutal for us teaching the ABE module. But my biotech class remains blocked, so I'm happy. So just, sorry about that, I got cut off, but you'll have access to my PowerPoint. So just a little bit about me. So I've been teaching. I've been in the district for about 27 years. I'm going into my 28th year. I used to be a math teacher at Rosemont, but I've been a AP Bio teacher for a long time, because Sam's only 22 years, right, Sam?
No.
Biotech teacher since '05, and I've been a coordinator for the Academy of Science and Medicine since '04, and I do I'm the Skills USA and HOSA advisor, and we just got back.2 who does Skills for HOSA? You guys should do it. It's really fun. And we just got back to 2 weeks ago. I took 6 students to Atlanta. This is like, I think this is our 7th or 8th year going to nationals now. And it was fun. There were 18,000 students, participants, so many different events. And we won. We won silver. Yay! Yeah, so, and that was fun. So I've been I still am a licensed clinical laboratory scientist for so 42 years as a working CLS. And I worked at USC for 32 years, and I just recently retired. So while I was teaching, I was still working. And the good thing about the hospital is that they're open every day. What is it said? It's 24/7, 365. So I worked on weekends and holidays. So my background is in microbiology and electrical engineering. And so that's why the ABE is important to me because of my background. So I've always lived since Sam was born, right? I've always lived, I've worked in a laboratory, So laboratory had been my life for many, and even now, because, I'm still teaching, so. I got one more year though, so. All right, so how do I actually integrate the ABE curriculum in my classes, right? So, all right, let's start with biotech. Does anybody teach biotech here? Okay, yay. So in 2000, and because Sam asked me this, so how did I find out about Wendy, right? Have you guys met Dr. Johnston, Wendy Johnston? Yeah. Yeah? Okay. So when I took over the academy in 2004, we were told that we needed a CTE component of the 10th graders. So then I picked something that was easy, right? Biotechnology, because before I started teaching, I was actually doing research. So I was doing a lot of molecular biology research. So I picked that. And then so the first person I needed to call was Wendy. And so Wendy said, take the workshop, okay, the Amgen workshop here. And so I did. So, and I've been teaching it since then. So thank you. Yeah. All right, so biotechnology, and sorry, do you use this textbook?
Not that one.
Okay.
I've used Ellen Doran's and then more recently the BioRad.
Okay. So yeah, so I use Ellen Doherty, and if you ever get a chance to do a workshop with Ellen Doherty, oh my gosh, she's amazing. She's in San Mateo. Sam, can I get some water? So what is biotechnology? It's my class. It's a year-long lab-intensive course, and it's basically very hands-on, very hands-on. So we do a lot of labs. So about the biotech program, it started in 2005. It fulfills the 10th grade CTE requirements of the Academy of Science and Medicine. Has been as ROP, a CTE class since 2005, and it became UC approved in 2006. And we're articulated with LA Mission College, and I put PCC there, a question mark, because for many years we were articulated with PCC, and just, I don't know, maybe 3-4 years ago, they just, every time I sent my renewal, it would just sit there and sit there. So then I just kind of gave up, and I said, done. So anyway, so hopefully, I'm sorry, where are you? What school?
I'm at Roosevelt High School in North Heights.
Roosevelt, okay. So LA Unified School, yes. Belmont. So it says here, this course was made possible by contributions from Dr. Wendy Johnston and crew from the Pasadena, from the Biotech Center, basically. And really, when I first started, when I called Wendy, she just pretty much gave me all the donations. She donated an incubator, donated a PCR machine, donated this, donated that. I mean, that's how we got started. Even a class set of micropipetters. So that's how I got started. So thank you, Wendy. So the Biotech Center provides equipment, materials, Saturday internship opportunities for my students. And then Amgen provides the training, like you guys are being trained now, so excuse me. Okay, all right, so what do my students learn in biotechnology? So they learn how to plate and culture bacteria, DNA isolation, gel electrophoresis, basically these are all from the ABE curriculum, right? Students are also exposed to column chromatography, so that's lab 6, and you guys will be doing that. PCR, I think you guys are doing the colony, yeah. I don't actually do that. I use the coastal marine, the DNA barcoding, so Yeah, DNA barcoding and bioinformatics, and of course, 4 weeks or to 5 weeks of ABE biotech experience. So we spend a lot of time doing this, so it's fun. Okay. All right, so my students also get to experience spending Saturdays with Sam, right, Sam, and the biotech PCC interns, or sometimes I think you guys have from LA Trade Tech and LAMC, right? So yes, they make reagents and pour plates. So some of the plates that you guys are using that are being shipped to the high schools, my students actually help in making those. So your biotech kids will probably be going there for their internship. They also spend like two and a half weeks at the stem cell module through PCC. We actually have a partnership through CERM, PCC, the CERM program. So they come to our school. So that's Mr. Santana, who's a professor, used to be Dr. Carol Liu, who would come to our classroom. It's kind of hard to see. Should we dim the light? Thank you, Sam. So this is my students actually performing staining stem cells, and that's he's giving them a lecture. So it's kind of cool. Presentations. The PCC interns come to my classroom, and then they learn about their research on stem cells. They spend like a day-long workshop. Not all of them though, because it's kind of selective sometimes. So maybe I'll send 3 of my students to PCC and work with. This past workshop, it was with Mr. Santana. So, all right. So we do about 4 weeks of the DNA barcoding. So if you ever get a chance to actually sign up to work with Coastal Marine Biolabs, do it. It's free. Coastal Marine Biolabs, CMB, right? So this barcoding is actually a collaboration with Coastal Marine Biolabs and University of Guelph in Canada. And so the goal is to create a database for basically, so we started out with fish when we started. We started out with fish, so now we're doing bees. So, and I was just telling Joe that it was kind of sad because just recently we did it and we only got, they send you 20 samples and we only got 5 amplicons. So those will then be sent to a sequencing place, and then they sequence it for us, and then they bring it back, and then we do bioinformatics. So if, and then they edit it, and then they get a barcode. They get a barcode, and then we upload that, and once it's uploaded, then the students get published. So that's really cool. So I really strongly suggest that you guys do that if you can. If you're not, there's also the BioRad kit that you guys can do. So, all right. So this is my class now. I have a 5-week summer class, and they just got done with Lab 6. So this kid is showing his RFP that he collected, his LE-lit. All right. So this is chromatography, column chromatography. So, anyway, so I have about 36 students. Okay, so this is what we cover. Okay, semester 1, what is biotechnology, and well, you see it. But here is where it gets really interesting. So chapter 4 is all about DNA and proteins. So this is now all, ABE. Second semester, I love it because it's basically, so if you look at chapter 8, it's a production of recombinant plasmid, right? And then bringing a biotech product to market. So what do we do after we have that plasmid? right? And so I'm just going to click here. So in 3.1, that's chapter 3, that's all about basic skills, okay? So we learn how to pipette, okay, so pipetting accuracy, but this is where ABE Lab 1 comes in, so the micropipetting, so it's perfect for it. And the 4.2, it's all about DNA, plasmids and bacteria culture. So I've written down like what labs, ABE labs, I can use to reinforce or to review, right? So even when I'm actually doing ABE Lab 2, then we go back and say, oh yeah, remember in 4.2, or concurrently, I could be assigning this too. So 4.3, 4.3 is all about gel electrophoresis, isolating and manipulating DNA. So again, Lab 1, but also Lab 4. Because Lab 4, ABE Lab 4 is about confirming or verification of your restriction digest, right? So again, great way to actually reinforce this, this unit or section. Okay. Chapter 5, now we go into the proteins. Proteins, amazing, right? 5.1, 5.2, ABE Lab 6, okay, perfect. And then, so I do an extension. Once we collect the RP, I do, we put it through PAGE, polyacrylamide gel electrophoresis, and again, that's perfect for this section, studying proteins. All right, in chapter 8, now it's getting really interesting. This is production of recombinant biotech data. And that's actually what you guys are doing, right? So you guys are doing lab 3 today, and that's ligation, right? So then lab 4 is basically confirming that you have the ligated, the recombinant plasmid. So what do you do after that? You do a transformation, right? And why do we care? Because we care because we need the proteins. Because as we say in biotech, right, DNA is flash, but protein is cash, right? So write that down and say it to your kids. Protein is cash, right? So that's when we do like the overnight culture and then column chromatography. Okay, that is when we actually isolate the protein. So what do we do once you have the protein? Well, you bring the product to market, right? So again, we talk about manufacturing, recovery, and purification. That's ABE Lab 6 again. And then there's column chromatography. And then I skip 10 and 11 because that's all plant biotechnology and, you know. Anyway, then I go to 12. I'm not a botanist. I'm a microbiologist. No. But medical biotech, this is where you talk about Amgen. What do they do? What are their products that they sell? So they're known for their Nepogen, their Aranesp, Prolia, Neulasta, Xgeva, I think that's how it's pronounced. So if you guys don't know these, I think we all know what I worked at Norris Cancer Hospital for 32 years. So Nepogen is a product that most of the patients use there, right? Because what is Nepogen? Nepogen stimulates your bone marrow to produce white cells, basically. And then the same thing with Prolia. That, to me, and I talk about it because just recently I was diagnosed with osteoporosis. So Prolia stimulates bone production. So yay. Then, of course, 13 is polymerase chain reaction. So again, you guys will be learning the colony PCR. I don't do that. I used to do the ALU. We don't do that anymore, right, Joe? Yeah. That was fun because you could put in genetics in there. So anyway, so I just do the barcoding. Okay, So, who are my AP Bio teachers? Yay! Woo! We all got our scores, right? Woo! I know. So my student just emailed me. He's like, I got a 5. That's the exciting part. Okay, so where does ABE fit in AP Bio? It fits in perfectly in Unit 6. Unit 6, that's gene expression and regulation, right? So, so we use Campbell, the 11th edition. So I apologize if you're not using that. But it's perfect for us because in chapter 16, it's all about DNA, right? It talks about plasmids and restriction enzymes and all that. DNA replication. So again, that is ABE Lab 2. In fact, ABE Lab 2 would be great to reinforce or to review during the AP, when you're reviewing for the AP exam. So chapter 17, gene expression. Okay, so that's the central dogma from DNA to polypeptide, right? And eventually a protein. So transcription, translation, again, that's ABE Labs 5 and 6. And of course, 18, it's regulation and gene expression, so both in prokaryotes and eukaryotes. So prokaryotes, we talk about the lac operons, the lac operon and the trip operon. So ABE Labs 5 and 6. Okay. And then this is the best part because chapter 20 is all about biotech, right? It's tools and biotech, biotechnology. So 2.1, okay, this is all ABE, right? So this is where actually, okay, we do 2 weeks of this. We just do, that's all we do. So, and during this time, my AP kids they're getting their reading assignments from chapter 20. They've already learned a lot about gene expression. So it's easier to teach them. So when we go to lab 2, lab 3, and by the way, and I just have to say this, but I'd say 70% of my AP kids are biotech kids, meaning they've already taken my biotech class. So when I say, yeah, so it really helps me a lot. And so I have 10 tables, 9 groups, sometimes 10. So each table, I make sure that each table has biotech students because then they already know how to micropipette, they've done these. So it really helps me a lot. So again, confirmation by gel electrophoresis. So anyway, it's perfect, right?
Orenda,
So
we have a question back here.
yes,
Oh, so do you do the same labs for biotech and for AP Bio?
exactly. Yeah,
Okay.
I did a complete series.
For
The
both?
only for both, different times though,
Okay.
right? Because oftentimes my biotech kids will come and help out in AP Bio to set up. The only thing that I don't do in AP Bio, just because because of the time crunch, is I don't do lab 6. So we do lab 1, lab 2, lab 3, lab 4, and lab 5. And I have the pacing guide.
For the AP Bio
Yes.
students that have already done biotech, do they enjoy doing the labs again?
Oh yeah, and what they enjoy is showing their skills. They go around and say, "Oh, I know how to do this." Yeah, but it helps me a lot because then I don't, because usually I have one. Usually my TA is a non-academy, non-AP, non-biotech. So it helps to have 2 biotech, ex-biotech students per table because they can help the non-biotech already. Yeah, so, yeah, it's great. So anyway, so now we go to our, I think I'm running out of time now. So let's go to our pacing guide, and this is where Sam is going to help me. So if we take a look at, and this is just me, feel free to look at it. So we do a lot. We do a lot. So we start out with the bleach lab, and I have a great example of a lab report from the bleach lab that you guys can take a look at, so if you want to use it. So that's semester 2. So Sam, can you go back to the first one?
Yes.
And I just want to go to the 4th week. So yeah, so now we go to, this is the first semester of biotech. We're starting to do pipetting, and then here we go. It says download the ABE lab manual, and then so they start working on. So here's the next week, and then ABE lab 2, lab 4, and so on. And I do a confirmation. Sometimes if you're hurting for time, I kind of do the transformation before lab 4, because lab 4, you could run it while you're screening, you're looking the next day after transformation, right? So that saves you 1 day. So anyway, all right. So moving on, scroll. Yeah. So now we go to semester 2, and scrolling. Okay, spectrophotometer. Stop. Go back up, please. So we do. Let's see, the biotech. We do biotech again. There's PCR, GMO. No, go back. Sorry. And it's just, I think, yeah. Okay. I'm not seeing it here, but that's okay. That's okay. So one more thing that I would like to say is that another way to reinforce this, or an extension of the ABE, is to do the biobuilder.org. Biobuilder.org. And this is it's synthetic biology, but it's genetic, and then you guys can talk about the difference between genetic engineering and synthetic biology, right? So if you go to the biobuilder.org, you guys can actually sign up to go to MIT. I was lucky enough to, when they first started, the very first workshop that was given by Dr. Natalie Coldell, I was asked or invited to go to MIT and learn about biobuilder synthetic biology. And then the next 4 years, I was asked to help kind of train teachers to do it. So, and the reason why I love it, because it's using electrical engineering principles to actually do genetic engineering. So, which is my background, right? Microbiology and electrical engineering. So it's perfect for me. And my students actually like it. But on top of that, they get to learn how to use a CAD program called Tinkercell. And so using Tinkercell, they can put it all together and simulate. So what they need to do is they need to, just like how you guys are learning lac operons, or operons, and you're putting they're modular, right? So, and then once you put it together, and then you put it in a chassis, which is a cell, and then you simulate it. So if your design is wrong, okay, your simulation will not work. So it's kind of cool. But again, it's a wonderful way to reinforce ABE. Okay, all right, next. And so this is my pacing guide. This is just summer.
Okay. You want to just do your block schedule?
Yeah. So the way we do it, because we have, I think it's 5 weeks for me for biotech. So we start out with, the lab manual is so amazing because there's a lot of information there. So we do, like the insulin story. But I also have a lot of videos that I'm sharing with you guys. And a lot of these are clickable, so they're hyperlinked. So we start out with practice and then an overview. Can you just click on that overview? So that overview is what you guys have been learning, right? So I get them excited about genetic engineering, what they'll be doing. things like that, and the end goal, right? So again, I keep reminding them that the end goal is cash, right? So anyway, I know, right? DNA's flash, protein is cash, right? So we start out with pre-lab, okay, with pre-lab, and they get reading assignments, and not just from the manual, but they're also getting the chapter from our textbook.
Is that from the biotech?
Yeah, from Ellen Doherty, yes. And then, so then the next day, so we're Tuesday, Thursdays, and it's block, and it's 2.5, and we're actually, it's nice, and it's gonna stay like that because we're biotech.
2.5 hours.
Yeah, so even when we go to regular schedule, yeah, my biotech class will because we're academy, we're CTE, we're special.
How were you guys able to lobby for that?
How what?
How were you guys able to lobby for that?
We didn't have
To
to.
make yourself known. Who was on your side then? Who had your back?
Well, we have funding.
CTE. has a lot of
CTE,
funding.
yeah, the CTE has a lot of funding, but we also, does anybody have an academy in their school? Yeah, so you know that we get, it's like 81,000 yearly.
But do they say that it has to be blocked? I'm just thinking like,
Oh, no, I was the one that asked for a block, because there's just no way you could do ABE labs with 55 minutes, right? Well, some people do. You're right. Some people do. But if you want to, spend a lot of time discussing it, so it would be your stretch. So anyway, you're right.
Is your summer class the 5 weeks? Is that 2.5 hours twice? a
No,
week
it's
as well?
5.
It's
5 hours every day for
every
5
day for
weeks.
5 weeks.
For
Okay.
5 weeks, yeah.
That explains why mine didn't quite work. Wasn't able to finish everything. We did
Yeah.
it 3 weeks for 5 hours each day.
Yeah, 3 weeks.
Yeah.
That's.
We didn't get to.
Yeah,
finish
that's
everything.
one semester
Yeah.
for us.
Well,
Or
that would have
one.
been. Well, I was trying to do pretty much the whole series in that time, but.
I just want to add that we do have a guest lecture series, a webinar series that's available to everyone. It's recorded, and we've covered several topics, which we'll show you on Thursday. And I just today thought of a couple of new ones that we should probably cover this year. So, Orenda, we're going to hit you up for help again.
I'll be retired in about a year, so you can hit me up anytime. Okay, so, but I spent a lot of time post-labbing. So when, in my biotech class, they do, like they do a flow chart, they read, and then, so when we actually do it, so can you click on that? Or actually, let's go to lab 2. I think lab 2 would be better. Yeah, so if you click on that, guys, again, I explain why we do each step. So scroll, please. So purpose, okay. What is a plasmid? Yep. Okay, keep going. Keep going. Don't stop. Keep going. All right. So again, this is a good way for you guys to review, reinforce, like what are restriction enzymes, what's HinD3, what's BAM-H1. BAM-H1, right? That's what I say. BAM. I get so excited because it's anything pertaining to bugs, microbiology. Okay, stop right there. So then I asked, we asked the questions like, why do we incubate at 37 degrees? And then what happens if the temperature is too high? So, yeah. So next slide, please. All right. Then you go to ligation, and then just keep going. Yeah, keep going. And then again, go back. And then this is a really good way to talk about, the different components or different parts of the DNA, right? So you have 3 parts with the nitrogenous base, the sugar, and the phosphate group. And so right here, okay, it's still, so when you ligate, this is still missing. So you got to seal the deal, right? You got to add the ligase. So now you have, you catalyze the reaction. So talk about that. So next. Yeah. And then, yeah. So again, a great way to review DNA. So Yeah. All right. So let's go to maybe lab 5,
Oh, okay.
the
Back here.
post lab.
Right.
Yeah, so a lot of these, the post lab, you guys can just click on it and then you get the lectures. I mean, feel free to use it. You don't have to use it. But anyway, so I like this one because we can talk about the Nobel Prize winning technique that you guys are using, right? Or doing. So, so if you see green, I just tell the kids, well, you guys are using red, so think red, right? But anyway, so yeah, next one. So Nobel Prize winning technique. Next. Okay, what is transformation? Next, please. Next. All right, and the different methods of transformation. Yeah, yes, ma'am.
So I'm noticing that, I'm just curious how your pre-lab is different from your post-lab discussion, because it doesn't have a lot of data analysis.
Sorry, say that again, please.
How do you distinguish between your post-lab and pre-lab discussion?
So the post-lab, Sam, can you add the questions? So the post-lab, no, sorry. No, you could go back to the same.
Okay, let me
Yeah.
go back. Okay.
So the pre-lab, I talk about the procedure itself, what we'll be doing, right? And then the post-lab, okay, next, please. Yeah. So this is just reinforcing, right? So here, stop. So these are now, can you go back to the previous slide? Yeah. So now you could ask these questions. Why did you do that? Okay. So what was a transforming solution for? What's in it, right? So it's more like answering some of the questions that they may have. Why did we use that? All right. So next. So and next. So there are questions as to why we did certain things. Next one, please.
So these slides are for both your pre-lab and your post-lab?
Yeah, so I don't do the questions when I'm pre-labbing. I just do the procedure. I do sort of like an overview and then the procedure, and then we do it, and then the following day, then I go over what they did as well as answer those questions.
I'd love to go over how you go over the procedures of that too.
Sorry?
I'll ask you more about how.
Oh, yeah, sure. But remember, I have 2.5 hours with them, so I have a lot of time with them.
Okay.
Go ahead. Next. Yeah, keep going. So these are just kind of going over their results too. So,
All right, that's the last slide.
yeah.
All right.
Yeah.
Moving back.
And then, so next slide. So you have, if you click on the lab videos, so these are just the videos that I use for each. So, and I've written down like which lab they're for, and feel free to use them. And I know that this workshop's gonna give you a lot of resources too. And you already have access to a lot of them. So, but it's just a good way for them to learn on their own. Okay, next slide.
Slide.
And then, yeah, paper plasmid, which is again a great way. So at the end of the complete series, you guys can use the paper plasmid to actually see if they actually understand what they did.
Afterwards?
Yeah. So it's an assessment. Yeah.
Sounds good.
Yeah. So next slide. All right. So these are just before we do the micropipetting, lab 1. I have on top of the pre-lab questions as well as the reading. Then I give them this worksheet to jot down the do's and don'ts of micropipetting. And then again, so the next day when we actually do it, then I kind of go over and then they have a list already. So this is a secret code. I think some of you guys use this. This is just, again, pipetting exercise. So you could do it before, or you could do it after, or you can do it as for fun. So anyway, next. And then, yeah, and then how to write it. Go back to how to write a lab report. So click on the PDF.
This one.
Yeah. So I used to teach at PCC with Dr. Pam Ebersole. So it was a Bio 102A and Bio 102B, and this is what we used. So feel free to use it, okay? I've actually summarized it, and I give it to my AP Bio. So can you click on the other one?
Okay. So this one?
Yes. So this is for AP Bio, so it's a little bit more, it's summarized. So if you guys feel free to use it, okay, it's there. But the most important thing is writing the conclusion. So if you just scroll to all the way at the bottom, so the conclusion should have, it's called REE, PE, and PA. So REE, discussing the results, okay, giving evidence, as well as detailed explanation. And then PE would be possible errors, and then PA, practical applications. And we end with an example.
Next slide, Brian.
Yeah, it's an example of,
Oh, next says.
example of the lab report.
Okay.
So this is our very first lab. Actually, our very first lab in biotech is making cheese, and we eat a lot of cheese, by the way. I always beg them not to cut cheese, all right? But eating cheese, making cheese, and eating fun. So flow chart, yeah, procedure, all right, and evidence, results, okay, analysis, coming up. And of course, the conclusion, the REE, PE, and PA, right? Okay. And then I think the last slide just the you have access to all the post-lab presentations. Ah. Yes.
Sorry, I'm backtracking a little bit.
No worries.
For AP Bio, I noticed that you were showing your pacing for chapters 18.
Yeah, 18, 19, yeah. Mm-hmm.
When you do the Amgen labs, do you do those labs separately as you're going over these chapters, or do you
Concurrently.
Oh, concurrently. So
Yes.
you don't do like this whole storyline, like all in a row. You just kind of pick it apart.
Well, I mean, we're learning about gene expression, right? So a great way to reinforce that is to do these labs.
Have you done both, where you do all the teaching first, and then like the 3 weeks of
Just not enough time, right? Remember, we got to get them ready for the AP exam. So, and especially this year, we were at the first day. So we lost 10 days. 10 days. If I had 10 extra days, yes, I would have. But you know, it's all about when the AP exam is. Right.
But site, my colleagues and I are trying to figure out if we should, because we've been doing like the straight 3 weeks of like Amgen labs.
Mm-hmm.
So we dedicate all January essentially to. So we're trying to like, hurry it up.
Right. But remember that, do you use Campbell?
Yes.
Okay, so chapter 20 is all about the tools, so 20.1, that's perfect for it.
So do you think, do you think it's better? I'm just trying to decide if we should sprinkle it across multiple chapters, or if we should do one.
I think it all depends on your, what you want to do too. But I think I just want to just get it done because the next unit is evolution, right? So that's why. But guys, 2 things I want you guys to remember when you're doing ABE is pack your patience, right? Lots of patience and flexibility. You need those 2 things. Very, very important. And if you can get helpers to help you set up, that helps a lot. I can say that I don't do it alone. So but Sam knows my students, so.
Yeah.
Anyway, thank you guys.
Thank you.
Yeah, yeah.
Okay.
Yeah, I want my